Well, we have Liz Parrish next. She, just like Bill asked me to sing his introduction, Liz didn't. And in fact, she said to make it very brief. And she doesn't need an introduction anyway. But Liz is a humanitarian, entrepreneur, author, innovator, and a public speaker for genetic cures. She's the founder and CEO of BioViva Sciences. As we know, BioViva is committed to extending healthy lifespans using gene therapy. They work on combinatorial gene therapies with its proprietary CMV gene therapy delivery platform. And let's welcome the wonderful, extraordinary, beautiful, remarkable Liz Parrish.
Yes. Welcome to the future. Welcome to your future. I hope that one day that all of you are a patient of my company. It's going to take a lot of work to get us all treated and get us all off to a much better start in decades of more lifespan. My company is BioViva. Yes, we treat biological aging with gene therapy. Our goal is to create a wonderful extended lifespan and health span, more importantly, for you. So the limit of your dreams, I think, is time, right? How many things would you do if you had the time to do them? If you had the plasticity of mind in order to keep all of your old memories and add new ones to it? Your value to the world is immense. It's actually invaluable. And I want you to understand that every day on this planet is an almost impossible experience. But we can make it even better by looking around us and looking within us at our genes. The difference between lifespan and health span, I think that everyone here is this is well understood. Today we live a long time by some people's mindsets, not by ours, but we don't live in good health for a long time. And so the first step, of course, in creating ultimate regenerative medicine and longevity is to start pushing that health span back, pushing it forward, rather, so that we live longer, better. And this graph is more than just squaring the curve, okay? This graph is a little bit grim because it's saying, what if we could live to the ultimate age of a human being and then just be done? Well, of course, we don't want to be done. But there's more to this curve that was pointed out to me than I understood before. If you look at ultimate health for humans today, of course, my company is focused on creating homeostasis in the cell so that we create a health span in which has never been seen in a human body before, because you are accumulating damage and you do have shorter telomeres due to cell division even when you're young. But if you look in that 20 to maybe going back to 15-year stage, that's when you're in optimal health and you're actually getting fewer returns on all the investment that you made in your early life. Dang it, that is not enough. By the time we can actually go out and have a great vacation and go do the things that we really want to do, we build our wealth and our network of friends, we have diminishing returns on our health. That's not okay. And aging is at the core of it, okay? So we understand that what is driving aging is causing the diseases that we die of today. These are the things that we want to push forward, push away, and eradicate. And I don't think that any civilized society at this point shouldn't be treating aging as a disease. Thank you. We do look at the hallmarks of aging. This is 9 hallmarks of aging. There's recently an expanded list, and as Bill Andrews said, this is a wish list, right? It gives us endpoints of which to pick off of and at which to target with cellular therapies. I just wanted to go ahead and get this out early in the talk because I got a lot of things to go to. Last year, but then this year in Journal, the data from my telomere length came out. And so anyone who wants to see that, that's peer reviewed. And we hope in the next 2 years to get all of my data out. And what's really exciting about that is that in between my first gene therapy, which some of you may not know I've taken, I have like 300 friends here that have been with me since the beginning of my journey. Some people here were actually here at the first talks that I did before I did a gene therapy in 2015 to push this type of technology forward, to get the first human data. And this is my telomeres since then. We do 3 types of telomere testing, 3 different companies rather, and this is just one of them. So my telomere length just came in a couple of weeks ago, and by this test it's over 9 kb. By life length it's over 12.7, and by a private lab it's around the same. So all of these testing companies have a little bit different measurements, but basically they all show lengthening of my telomeres since my first treatment. And then in 2020 I had a second treatment that we added clotho and PGC1 alpha on top of the follistatin and the telomerase gene therapy that I had taken before. Now, the reason that I wanted to tell you about my gene therapies is because since the inception of my company, I have taken 4 different genes. Okay, and they're all considered geroprotectors, or they treat a hallmark of aging. Now, one of the caveats that we have in gene therapy is actually getting these genes into the cells at one time. So along the road of trying to get patient access, we wanted to create a gene therapy delivery method that could get gene therapies into your cells at one time, a bunch of them, like a mini artificial chromosome that would help you live longer. And we are also interested in working on precision medicine. So in the gene therapy space, there are now 9 approved gene therapies for monogenic disease. So if you're born with a rare disease, there are gene therapies in the pipeline for you, but there are no gene therapies yet in the pipeline through the regulatory system for aging. So we created a system in which we could give you your gene therapies around aging and add a gene therapy for monogenic disease. And this is a little video about it.
Gene therapy is a new and exciting class of medicines that can revolutionize patient treatment. BioViva's gene therapy uses CMV, where harmful viral DNA is replaced with potentially beneficial therapeutic genes. Vectors are administered to the body and are used to enter the patient's cells and transfer the beneficial genetic payload. BioViva focuses on genes that fight against the deleterious effects of aging. Genes are made of DNA, the blueprints for your body. Most people have 2 copies of each gene. These blueprints make enzymes and proteins. They are responsible for eye color and athletic ability and affect our long-term health outcomes. Aging is cellular degeneration over time, and we see the effect in our skin and in all the organs of our body. By treating cellular aging, BioViva hopes to reduce and even reverse this decline. After BioViva's gene therapy is delivered, the patient's cells act as factories producing the gene's therapeutic proteins. These proteins are designed to treat aging at the cellular level. Gene therapy works by enhancing the body's natural biological ability to fight disease by enhancing the cell's ability to create the proteins it needs within the body. CMV can carry more genetic payload than traditional gene therapy delivery methods, allowing BioViva to treat complex diseases such as aging. With BioViva's CMV vector, the company can deliver larger genetic payloads. Bigger payloads mean more genetic material, and the potential to deliver multiple genes at once, creating a better precision medicine effect. From lengthening telomeres that shorten as we age, increasing muscle loss that is incurred with aging, to adding geroprotectors and cellular reprogramming factors, BioViva's CMV gene therapy delivery platform will be with you every step of your longevity. BioViva's CMV gene therapy shows low immunogenicity in studies and can deliver multiple genes in one treatment, potentially treating complex diseases with a lower dose. Intranasal delivery and redosing were shown in animal studies. Multiple organ delivery shows the potential for targeting the whole organism. BioViva's development of the CMV gene therapy delivery platform targeting age-related decline will revolutionize millions of people's lives, helping us live longer and, more importantly, healthier lives. Visit www.biovivascience.com.
So our goal is more genetic payload, obviously. I think that video said that about 5 times, into the cell with a predictable lower dose treatment. So instead of having to do 4 or 5 different gene therapies, you would only have to do 1. So in, let's see, was it May of this year, our paper came out in PNAS describing what happened in our mouse study. Now, if you talk to me, mice are not humans, and I'm not thrilled about mouse studies, but this is where we do have to start in order to get through the regulatory system. In this paper, we used 2 genes. We used in one arm of the study the follistatin gene, and we used it in 2 ways, intranasal and intraperitoneal, which sort of mimics an intravenous injection of gene therapy. In the second one, we used the telomerase reverse transcriptase gene, the gene that Bill talks about that lengthens the ends of the caps of the chromosomes. So what happened? We saw increased lifespan and health span in these mice. So on the far right of the screen, you'll see the untreated mice on the top. They were all about 24 months at this point. The telomerase treated mice in the middle, and just to let you know, you can read the graph, but the telomerase treated mice performed best upon every test measurable. And then the follistatin mice on the bottom right, and those girls had a lot of muscle mass. Follistatin is a myostatin inhibitor. It muscles you up, and it's great for metabolic health, as you see on this panel. One of the great things that somebody could see maybe translated in their life, better hair, better mitochondrial function, muscle mass, better metabolic markers, and faster response times. The TERT mice were 5 times faster than the controls. So again, mice are not humans, but this is the goal of my company, to get these drugs to you. If this data translated directly to humans, and you were a human that was only going to live to, say, around 80 years of age, with telomerase, you might live to be older than a supercentenarian, over 110. And with the follistatin alone, you would live to be about 100 years old. And those are massive gains, because this is health span and lifespan. And what we're trying to do is create therapies, low-hanging fruit, gene discoveries that have been in research and development for over a decade, that have never translated to humans, that have been reproduced in multiple labs, and actually do them justice, get them to humans. So what's next? Right now we work with Rutgers University. George Church is an owner of my company, and he oversees all of our research. And his interest is combinatorial gene therapies, as is ours. And so we are in the combinatorial gene therapy into human cells, part of the research. And then we're also going to have to do another mouse study for the preclinical work. So we're using 4 genes. Again, we're using telomerase reverse transcriptase, lengthens the caps, the ends of the chromosomes. Clotho is a geroprotector, and also associated with better cognitive health. So a lot of people reach out about that. PGC1 alpha, that's a gene that you upregulate when you work out. So it's associated with working out and better mitochondrial function. And follistatin, which increases your muscle mass and combats against metabolic health issues. But it's not enough to just have this technology. And again, that's my company's job, is to take the big risk, to really get out there, to get the good and the bad media, to get it all, because the most important thing is that this technology gets to you. We are going to do a reproduction of the dementia study. In 2020, 5 patients were treated to free gene therapy to see if we could actually combat the deleterious effects of dementia, an absolutely insidious, horrible disease. In this study, these patients, I have to preface this with, it was not a cure. So, but what we did see could be absolutely life-changing and get people farther along to a cure, okay? So we used telomerase reverse transcriptase, and we used Clotho Telomerase reverse transcriptase is associated with the removal of tau tangles and better outcomes in cellular health. Klotho is associated with the removal of some beta amyloid plaques, but more importantly, cognitive health. So people upon autopsy that they found had Alzheimer's after they died, that's really how it's fully diagnosed, who didn't suffer the deleterious effects of cognitive decline were Klotho upregulators. So we're hoping that this helps. So what did we see? We saw an increase in scores in the Folstein test. Now, predictably, people with dementia, they actually decline. So they just decline at a slower rate with interventions. We saw an increase in cognitive benefits, and we're going to continue to follow these patients. And we saw a calculated biological age betterment through telomere length. And again, since this is all experimental therapy, this was unexpected. This treatment was done intranasally into patients, and it was a small dose of telomerase reverse transcriptase and Klotho. That we saw telomere lengthening in the T lymphocytes was actually a shock to us and was not something that we thought would be an endpoint of the study. I want to tell you that the coalition is actually helping to raise money to see if we can reproduce the study, but we don't want to just reproduce it. We want to go for a higher titrated dose. We're looking to knock out what is actually causing dementia, and we think that we're on the right track. We're going to see if a higher dose does better, and if not, we'll start adding new genes into the newer constructs. So if you're interested in supporting this, please do. You might even be a patient of the study or someone that you know. Again, it's the coalition that's taking care of this. It's not directly through BioViva, and we are so happy to have their support. I just want a really quick plug for 30 seconds, the Code Keeper. People ask me all the time what is happening in gene therapy, and we used our own money to put this together for you. This is a database that has everything happening right now in gene therapy. If you don't find something in here, then you need to contact us through the scripting system, and it will contact a PhD, which will look up the paper in which you have pointed out is not in the database. This will tell you everything in your disease that's happening in gene therapy. It'll tell you the animal model that it's happening in so that you can actually brace for your expectations. And if any of these research groups have something that would be beneficial to you, maybe you want to help fund them or get out and pound the pavement and turn this into a translated medicine into a company. My whole start in this was my son being diagnosed with type 1 diabetes. I was not a longevity person. I wasn't trying to live forever. I was trying to translate cures to kids. And by pounding the pavement and looking and seeing what was happening in research, I realized that not only could we help these kids live longer and find genetic cures for kids, but we could help billions of people live longer too. So don't underestimate yourself. Yeah, this is really about you, okay? Because that I'm here is a very strange thing. I'm just meeting an unmet need, and you can do that too. This is our exceptional team of players. We have a gentleman that's been at Merck for over 10 years working with CMV, the immunization for CMV. We have Bill Andrews, of course, a specialist in telomeres, Matthew Schultz, who's had several successful companies in biotechnology, and of course I owe a lot to one of our co-founders, George Church, because he's a driving force behind the company. I also have to thank Hua and Dabu at Rutgers University. They're specialists in CMV, and they're the ones who do all of our research and development. They're just fantastic. I wouldn't be anywhere without Aubrey de Grey, and John Schlondorn is just, if you don't know him, you got to look him up. Now, I'm going to spend the last few minutes talking about something that's near and dear to my heart. This is not a product of BioViva. This is a product of patient advocacy. This is really the only place where I have any roots and bearing to speak. I'm not a scientist. I'm not a medical doctor. I am merely trying to translate medicine to you. In the United States today, the drugs that you are taking are at minimum over 15 years old. Drugs are working at a very slow pace. Regulation has slowed almost everything to a halt. You can't treat biological aging. To use a gene therapy that might help everyone in this room, we have to find some endpoints in a certain disease and treat diseased people and see if we can actually, as secondary endpoints, treat biological aging. And because we're cutting edge, sometimes the regulatory system won't even deal with a whole bunch of companies that you've seen speak here. This is inhumane. We are demanding a humane route to drug development. We need everyone who is terminally ill or has a disease that has no cure, which is pretty much everyone in this room. We need them to have the right to choose their own medicine. We're asking for a regulatory route that sits outside of the FDA. The FDA should not be accepting mouse data. Mice are not indicative of how a drug will work in a human. They should be accepting human data from a platform like Best Choice Medicine. We would already know whether a drug would be a success or not. Did you know that if you're over 80, you can't even get into a clinical trial? Your DALI and QALYs are already outside of their preliminary assessment of what is okay, even if you were a person who might live to 100. The system is ageist, it's unacceptable, and it's crushing small companies that cannot afford to pay for this high regulatory cost that doesn't get you a safer drug. The FDA, the FDA has approved 10 of the most dangerous drugs, and that's outside of the opioid crisis. We're asking for a new system in which not only biotechnology companies, but universities could step up with their basement full of patents and start translating all of that research to humans where it belongs. This is the first aging war. We have to stop sending our children to wars. We need to start valuing life and letting people have the dignity to try drugs. In my paper for my thesis, which boiled down into a paper that Bill Andrews and I wrote and had peer-reviewed, we found that people had a better appreciation for life if they were given the dignity to try drugs that may not even work in them. Because that is what it comes down to, is purpose. And we believe that today's biotechnology is good enough to have an impact, that we will in fact be able to target these diseases and hopefully get these people to the whole next stage to see a cure for aging. Anyway, and finally, because I've run a little over time, the BCM plan would give the people the dignity to try drugs sometimes decades before they would get through to a regulatory system, and in many cases, not to a regulatory system at all. The 4 gene therapies that I took should have been available to humans in clinical trials years ago. I am not against the FDA. I am not against safe drugs. We have just had the inability to actually create them, and I want to be part of that. So thank you so much.
Thank you. Thank you, Liz.